Assalamualaikum, and thank you for Dr. Ramisah for teaching ours in Gtn3113. First of all, I'm Nur Nakiumandi Adomanap, which number matric S56523 will lead our topic topic is challenge in conversation on farming genetic resource. We all are from group 4 that include group member which is Nur Syahanin bin Syahidan, Iza Inani binti Muhammad Yusof, Nur Liana binti Sayuti, and Nur Syuhada binti Piramdi. Our issue is included: first, gene bank management; second, conversation perspectives in situ and ex situ; third, the management of facility; fourth, the long and short term storage of field crops; and the last one is the conversation need in physiology and allied requirement.
Gene bank management. A gene bank is a type of biorepository that is used to store specific genes, and then gene bank manage genetic resources and information about those resources. And agriculture system require a diverse type of genetic resources to improve production, nutritional qualities, and a long-term reliability. For gene bank management, envelope 3, facilities, collection and management of information. For management facilities, responsibility for operating the gene bank and exercise facilities. It is a facility for the maintaining crop diversity. For management collection, we divide by two, strategy and techniques. For strategy, the mandate of the gene bank and the collection mission objective influence the sampling approach, such as collecting certain genotype on a target strategy and minimizing genetic diversity losses due to genetic erosion. Techniques. The collection technique and equipment used will be determined by the type of material to be graded. For instance, seed, pollen, tissue culture, vegetative propagules, field gene bank, and so on. The samples should be of the same type of the dose used by the farmers. For the management information, is generated by the gene bank. The parameters on which this information is recorded are the management descriptor.
Next, conservation perspective in situ and ex situ. In situ means the preservation of variable population in their natural habitat, which is a dynamic system that permits biological resources to adapt and alter through time through natural accumulation. While ex situ is the preservation of genetic materials outside the natural environment, which the species arose with the purpose of preserving the material's genetic integrity at the moment of collection. For example, botanical garden, field gene bank, and seed gene bank. Then for the several issues in situ, such as cost, size, maintenance issues, and the risk of genetic wipeout as a consequence of natural disasters such as fire and so on. While for ex situ, it faces the danger of germplasm loss due to disease, stress, or disaster, and a great amount of space and labor are necessary to preserve a tiny fraction of the variety. Okay, so there are several strategies related to in situ and ex situ. For in situ, we monitoring the location, so we protect and taking any action such as policy improvement aimed at tourist exploitation and the possible detrimental influence of animals such as rat. Next is eco-development, which is a program permitted eco-development involved of biological diversity that include local communities has been initiated in recent years. So it can contribute to the survival of all native species and subspecies. While for the ex situ cryopreservation, which is the material may kept in small low maintenance refrigeration units for an extended length of time. However, in Malaysia, these strategies are exclusively used for certain reasons. So I believe they should begin to use this method to promote ex situ conservation. Next is monitoring as well. Our botanical garden, freezing bank, since it took years to keep them in a good shape.
Management of facility. Finally, the gene bank manager is in control of the gene bank and its accompanying facilities. It is critical to operate the property effectively and to make effort to maintain long-term security and cheap maintenance costs. Collection costs may be kept low by selecting appropriate equipment based on the number of collect to be held. Refrigeration equipment is expensive to operate and can be difficult to repair at times. To assist overcome the present collection vulnerability to voltage fluctuation and equipment fails, standby copy of important refrigeration equipment, voltage regulator and backup generator are advised. When analyzing the security of gene bank facilities, organization continuity, natural disaster, the stability of the local power supply, and the availability of a backup storage location should all be taken into account. Engineer and maintenance staff must be meticulous in their jobs. Temperature and relative humidity level should be recorded and monitored in the medium-term cool storage and drying chamber. Management is in charge of the security issues that must be dealt with all the appropriate levels, such as testing, to ensure that cooling, safety and backup systems are operational.
Long-term and short-term storage of field crop. First of all, we must know the type of seed, that a kind of variance of variety. This is included the taxonomy of the plant that we want to gain the seed, like and the hierarchy of the plant that included the order, family, species and genus. Other than that, we must know the lifespan of the seed can be stored. For example, we just have now the long term of storage of seed is kind of orthodox seed, while the short term for storage is the recalcitrant seed. From the storage, that seed must undergo the desiccation or known as drying tolerance is required on all of the seeds. This desiccation tolerance of plant can show the system of biology perspective on molecular and genetic pathway. That means that can be the dry seed can be grown as a living organism. Follow the Biodiversity International that we must have a best storage condition. That means the atmosphere must be good, the equilibrium of humidity must be better, and they must store in the low temperature. For storage, we can choose the cryopreservation. That means the cryopreservation is the process to prevent the structures intact living cell or tissue by freezing them in cryogenic temperature. Okay, this means this convection freezing and vertification technique that be used to role playing the tissue and the organ to achieve the cryopreservation. Cryopreservation of the tissue created by product in a circular to achieve the ready to use requirement in the clinical application and the regenerative medicine, especially in the rapid advance of a tissue engineering. Cryo storage must be necessary in a short time to avoid ice crystallization. Method that they apply in cryopreservation is the temperature must be below 15 Celsius. That the minimum of the temperature must be lowest than negative 20 Celsius. For moisture content, follow the Frenzy in 2011 that the orthodox seed must lose the moisture content in 3% until 7%, while the recalced seed must lose than 30% until 70% lost moisture content. We can that ABA can control the production of unsaturated late embryo protein, that LEA, and thermostable heat shock protein SHSP, and the non-reduction oligosaccharide.
Okay, then I will talk about the maintenance, why we should maintain of seeds under good conditions. The first one is we want to loss of viability in storage is associated with the considerable amount of genetic change. Thus, generation should be undertaken as viability has begun to fall significantly. Then, the cost of regeneration are high, and the difficulties and danger are considerable. Moreover, loss of purity can occur through mechanical mixing and cross-pollination. And in generally heterogeneous sample, selection can occur depending on environmental conditions in which the generation is carried out. Last but not least, the capital and running costs of providing really good conditions are not much greater than for providing storage condition of inferior quality. Also, storage at the preferred standard require less detailed management than thus providing pure storage.